In this video, we will discuss about how this isoferulic acid suppresses proliferation and metastasis in pancreatic cancer cells. So far, we have understood that isoferulic acid triggers intrinsic apoptosis by damaging the mitochondria of the pancreatic cancer cell lines, and these things we have understood on the, or these things are evident by checking the effect of isoferulic acid on PANC1 and BXPC3 cell lines, and we have used, or the researchers have used, JC1 aggregates analysis to confirm the healthiness of the mitochondria when they are treated with isoferulic acid, when cells are treated with isoferulic acid. So in this video they wanted to check whether the big picture of isoferulic acid, that is to suppress proliferation and metastasis in pancreatic cancer, occurs or not. So first, they did it using three different approaches. First one is this EDU test, and this EDU test is works in a way that when the cells are proliferative in nature, it will give high signals, and when the cells are non-proliferative in nature, it will give low signals, as this will usually go and bind to the DNA. The cells need to be fixed and permealized. So in the control cell, we can see that the signals for the EDU is higher compared to the case when the cells are treated with, or the PANC1 cells are treated with isoferulic acid. And with increase in the concentration of isoferulic acid, we can see the decrease in the signals. So this indicates that isoferulic acid suppress the proliferations of the pancreatic cancer cell lines or PANC1 cells. And similar results they have observed for BXPC3 cell lines also. That is, when the cells are treated with isoferulic acid, so with increase in the concentration of isoferulic acid, they see a decrease in the EDU signals, indicating that the proliferations of this pancreatic cancer cell lines goes down. And after validating their hypothesis using the EDU assay, they also wanted to check this hypothesis using PCNA. PCNA also works in a similar way. That is, when it expresses higher in the cells which are proliferative and metastatic in nature. So in the controls, they can see that the amount of PCNA is higher compared to the case of when the cells are treated with isoferulic acid. And with increase in the concentration of isoferulic acid, the PCNA amount also goes down. And but the control, we have used beta-actin, and the control signals remain same in all the cases. And when it comes to the BXPC3, they also see the similar trends in the BXPC3. That is, in the control, the signals of the PCNA is higher, whereas in the other case, that is when the isoferulic acids are low, the signals are when the isoferulic acids are when the cells are treated with isoferulic acid, they see a decrease in the PCNA signals, and with increase in the concentration of isoferulic acid, the PCNA concentration goes down, whereas beta-actin amount remains same, which is act as a control. And finally, they also did a wound healing assay, where we can see that in the case of control, after 48 hours, almost most of the wounds are covered. But when the cells are treated with isoferulic acid at low concentration, at this point we do not see too much difference. But when the concentration of isoferulic acid increases, the wound healing capacity of the cells goes down, or the proliferation capacities of the cells goes down. And this capacity further goes down when the isoferulic acid concentration increases. So this indicates that the proliferation capacities of the PANC1 cell goes down with increase in the concentration of isoferulic acid. And when they check this wound healing assay in the BXPC3 cell lines, they also find the similar results. And in this case, they can see that the difference between the control and isoferulic acid at low concentrations also, which is quite evident. And when they increase the concentration of isoferulic acid, they see a further decrease in the wound healing capacity, and this decrease in capacity further enhanced when the isoferulic acid concentration increased to a 12.5 macromolar. So all these three assays in together indicates that this isoferulic acid suppresses proliferation and metastatic properties in pancreatic cancer cells when And when they are treated with so this isoferulic acids, let me repeat the statement, this proliferations and metastatic properties of the pancreatic cancer cell goes down when they are treated with isoferulic acid. And we have also seen in the previous video that uh, it has happened because of the intrinsic apoptosis, which is triggered by weakening the mitochondrial membrane, and the cytochrome C will then move into the cytoplasm, and that will cause apoptosis. I hope you understood their how they validated their hypothesis. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.